regulators of gene expression. And it's going to be the roles of these regulatory proteins, and then in a bit, the roles of some of the host proteins that mediate this interplay between the virus and host, and probably are the principal determinants of the course of infection. Because after all, after an HIV infection, it might be a year or so. Dr. Sears gave you this slide on the kinetics, but a year or so of the acute infection, then there's seroconversion, then you go through several years of chronic infection before the syndrome, AIDS, develops maybe 8 to 10 years later. All right, so how do TAT and REV work? Because TAT and REV, kind of the global outcome of their function is to increase expression. They're positive regulators to increase expression of the HIV genes that are going to be important to make progeny virulence. So TAT, the protein TAT is the viral protein, and it is produced from multiply spliced RNAs. So again, when the cell Pol2 transcribes our integrated provirus using the viral promoters and enhancers, we get this primary transcript that is transported to the cytoplasm to make GAG precursor. It will also make the GAG Pol fusion precursor as a result of that frame shift. Singly spliced RNAs generate the envelope precursor, but then we get these multiply spliced RNAs that make the regulatory proteins, including TAT. So TAT is made from a multiply spliced RNA, and TAT gets its name. It's a trans activator of transcription. TAT is where the name TAT comes from. And so TAT increases HIV transcription, and it does so by stimulating Pol2, the cellular RNA polymerase 2, but it stimulates elongation. And so the study of TAT actually told us a lot about how cellular Pol2 works in the elongation mode. All right, so how does this work? So TAT, the protein TAT, works through a target RNA. So the target of TAT is an RNA sequence. All right, so we have our integrated provirus. The provirus is integrated, and we have our promoter and enhancer to which Pol2 binds to transcribe the provirus to make the viral RNA. So Pol2 binds, and RNA polymerase 2 has what's called a CTD. So a CTD is the C-terminal domain of RNA polymerase 2. And when RNA polymerase 2 is in the initiation mode of transcription, it's dephosphorylated. But for RNA polymerase 2 to really work efficiently in elongation of transcription in a eukaryotic cell, as you learned in genetics, in the elongation mode, it's phosphorylated. It's hyperphosphorylated, lots of phosphates, on the C-terminal domain. And when that happens, then Pol2 works really well in elongation. So how does this happen? So Pol2 binds to the DNA, where the provirus is, initiates transcription, and as we make some RNA, that RNA has a sequence, that's called TAR. So the target RNA, the sequence is TAR. So TAT is going to bind to TAR. So this would be the TAR sequence as we begin to transcribe the gene. So then TAT binds TAR, and then when TAT binds TAR, this then leads to the recruitment of the kinase. So the kinase is CDK9, cyclin-dependent protein kinase 9. And this kinase is regulated by cyclin, and so it's cyclin T. So from this multiply spliced RNA, we make a little bit of TAT protein. This TAT protein then is targeted back to the nucleus, where the cell polymerase is trying to transcribe the viral provirus. It's integrated. So we get a little transcription, and among the RNA sequence of that HIV transcript is this TAR sequence, and that the TAT protein binds to, which recruits the kinase and the cyclin that makes the kinase work. This then leads to the phosphorylation of the C-terminal domain of the polymerase. So we put those phosphates on, and then that really activates the polymerase, so we get a lot more transcription where we're looking more. So anyway, when this phosphorylation occurs, then we're in the elongation mode, and we get way more transcription of the provirus. All right, so that's how TAT works, to basically give a more robust production of the viral RNAs. Rev is another regulatory protein that's made from multiply spliced RNAs. So a different splicing, a different splicing pattern then gives us the transcript that makes the Rev protein. And Rev gets its name as regulator of virion proteins. Regulator of expression of virion proteins, REV. So how does Rev work? So Rev, like TAT, is an RNA binding protein. So the Rev protein binds to the viral sequence RRE. So RRE is an RNA sequence that's present in many transcripts. And the RRE, many of the viral transcripts. And the RRE stands for Rev responsive element. So that just means that an RNA that has RRE into it, the Rev protein can bind. And the consequence of that binding of Rev to RRE is to Rev binding to RRE leads to an increase in transport of the viral RNA from the nucleus to the cytoplasm, so it can be translated. Okay, so what RNAs have the RRE element? The RNAs that have the RRE element are the unspliced RNAs. So they're going to make the GAG precursor, and they're going to make the GAG pol and then singly spliced. And so these are the RNAs that make the envelope protein. So these are the structural proteins, the virion proteins. So Rev increases the production of the proteins that we need to make progeny virions, and it does so by increasing the transport rate from the nucleus to the cytoplasm of unspliced and singly spliced RNAs that have that RRE element. And that happens because the complex interacts with the protein exportin 1, which is the cell protein machinery that you need to transport an RNA from the nucleus of the cell to the cytoplasm. Okay, so TAT and REV both are regulatory gene products of the virus that increase multiplication by increasing expression ultimately of our components that we need to make progeny virions. Okay, so as a result of that, then we make the structural components, the nucleocapsid components. The envelope proteins are made and inserted into the plasma membrane, conceptually similar processes that we've seen with other viral glycoproteins. The nucleocapsids then that are formed interact with the regions of the membrane that are altered with matrix and glycoproteins to trigger then the budding of our progeny HIV virions from the infected T cell, and so we produce progeny. So that's how the virus replicates, and these are the two major positive regulators. Okay, how does HIV spread? So in the context of spread of HIV, how do we increase the number of infected cells in the population? So we've just gone through the replication of HIV to produce progeny virions. So in terms of spread of HIV within the host, spread in the context of increasing the number of infected cells in the system. So how can that happen? So there are three conceptual ways that this can happen. The first is through infections with free virions. So basically the scheme that we went through, we infected with a virion of a T cell that had the CD4 surface receptor and the appropriate co-receptor. So that's one way to increase the number of infected cells in the host, is simply by standard virology, you infect with an infectious virion. A second mechanism is through cell-to-cell fusion. That is, if you have an infected cell, and much as GP41 mediated the fusion of the viral envelope cell's plasma membrane, if you have an infected T cell, it can fuse with an uninfected cell to effectively then transfer the virus from the infected cell to the uninfected cell through this syncytia that's formed. So cell-to-cell fusion, you get in infections what's called syncytia, which basically are multinuclear masses of cells because you've had a fusion of one cell with the next. Yes?
Now, is this mediated by GP41?
Yes, that's the fusogenic activity. And actually, it's the basic principle that we touched upon when we talked about paramyxoviruses, because a very old technique in somatic cell genetics, where sometimes fusions were made to study sort of what gene on what chromosome causes what event, was to make fusions between two different types of cells. Even a mouse and human cell you could fuse together. And that was done by using the fusion activity of a paramyxovirus. So you inactivated the virus so it wouldn't replicate, but it still had the fusion activity. So HIV has a fusion activity too. GP41 mediates the fusion. All right, so that's the second route. The third route of increasing the number of cells is when the cell that's infected goes through cell division. Because the provirus is integrated into the chromatin, the cells divide, each of the daughter cells will be infected. So, third mechanism would be integrated provirus that's integrated into the chromatin. When you have cell division, then the daughter cells, of course, get provirus, so you increase the number of cells. All right, so the virus then has developed these gene products to enhance expression, and we've had pretty efficient replication. How about on the host side? Because the course of an HIV infection goes over several years before the syndrome of AIDS develops. So what's going on the host side? So there are various cellular restriction factors that have been identified. And many of these factors are part of what we call the innate immune response, in that some of them will be inducible by interferon to increase the concentration of the factor in the cell to limit the replication of the virus. So they serve effectively, or can serve as the powerful restrictors of HIV replication. So the virus, though, has evolved strategies to inhibit these cellular components. So what are some of the components and how do they work? One of them you considered in section, and that's ApoBec. What the ApoBecs are a family of deaminases. There are 7 of these deaminases, and they deaminate cytosine to uracil. So if you catalyze a C4 deamination of a C, you get a U. So a U is not a C, of course. And so among the deaminases, the first of the deaminases that were discovered was ApoBec1, which was actually an RNA deaminase. Important when you look at the metabolism, remarkable. But then there are others that were discovered, and among them are ApoBec3G, which is the main one, but 3D, 3F, and 3H. And these ApoBecs have anti-HIV activity. And so what happens, as you learned in section, is that they're encapsulated into the virion. So when HIV replicates and this progeny virions are formed, ApoBec3G and the others, 3, can be encapsulated into the virion. So the virion has that cellular enzyme with it. And so then, when the virion infects a new cell, during the process of reverse transcription, when you're making first strand DNA, that first strand DNA gets deaminated. So you have C to U deaminations occur. And then when second strand DNA gets synthesized, you end up with the effectively G to A mutations. Because you don't have a C, you have a U, so U would give an A instead of a G, and the second strand is there. So this leads to mutations in the viral nucleic acid that then ultimately lower the replication of the virus. So the virus has evolved to block this system. The virus makes the protein VIF. So VIF stands for viral infectivity factor. That's where the name comes from. So VIF inhibits the APOVEC. So the viral gene expression makes this accessory protein VIF, the viral protein, that inhibits the cellular innate component to down-regulate that activity so the virus can replicate better. So APOVEC is one of the cellular restriction factors. Another is called tethering. And tethering in the initial literature was called BST2. It has a couple of names. So tethering is a name I'd like you to remember, and it kind of goes with the function. So the tethering protein essentially tethers the progeny virions to the host cell. So when the virions are being produced and would normally bud, they're not released from the cell. They're stuck to the cell by virtue of the interaction between the virion and BST2 or tethering. So tethering restricts release. So the regulatory protein VPU that's made by the virus blocks tethering, which then facilitates release. So it's another example of the interplay between the virus and host. And the balance of all of this then is going to really determine how much virus we produce. And then a third one of these restriction factors, more recently identified, is called SAM HD1. And this cellular protein is a phosphohydrolase. Phosphohydrolase. So as the name implies, it hydrolyzes phosphates. So it's a deoxynucleotide triphosphate phosphohydrolase. So the DNTP pool in the cell is depleted. And so you need deoxynucleotide triphosphates because they're the substrate for reverse transcriptase when we're making the provirus. And if you deplete that pool, it limits multiplication. And so this phosphohydrolase then catalyzes the dephosphorylation to get the deoxynucleotide and the phosphates are hydrolyzed off, so you deplete the pool. All right, so those are some of the examples of restriction factors that modulate the efficiency of replication of the virus in the infected cell. So where are we in terms of disease then, related to HIV infection and progression to AIDS? What are the views? And so in terms of epidemiology now, it's estimated that in the US, there's in the order of 1.2 million people that are living that are infected with HIV. Globally, estimates are in the order of 35 to 40 million people are infected. So the bulk of these cases, and I gave you a figure on your handout, it's a little bit old, but it still is accurate in terms of the distribution globally. The bulk of the cases are globally are sub-Saharan Africa, where it remains a huge problem. Current estimates are greater than 25 million people infected with HIV in sub-Saharan Africa. The other major area is Asia, and the estimates are in the order of 7 million people in Asia now are infected with HIV and living. All right, so where are we at? And what's the situation in the U.S. and what's the situation for Italy with this threat? So this slide illustrates where we've come. And it illustrates the period of time. The statistics are from Atlanta, CDC. And so what this shows are the death rates in males aged 25 to 44. From the period early 80s to the mid to late 90s. And so I took this period because it reflects the period from when the virus was discovered in the early 80s. Of the disease that we will always recognize by the physicians and the virus started shortly out. So in guys, the major cause of death is typically is you can't see the scale, but it's accidents. So for males, of course we're invincible, nothing will happen. But things do happen. The major cause of death is accidents. And the rate from accidents and injuries of death is in the order of 50 to 60 deaths per 100,000 population in the U.S. And that's remained relatively constant over time. And then the next is heart disease, suicide, cancer, and murder. As 4 causes of death. And you can't see the number, but the number is in the order of 25 to 30. This is 30 years of this time. So heart disease, suicide, cancer, and homicide. The rates of 25 to 30. Then what you see is the red line, the only curve that really changes, and that's death from HIV infection. So we went from the time of recognition in the early 80s to the mid-90s. HIV infection became the major cause of death in males in the U.S. and the U.S. And then it plateaued and went down. So we talked about life decrease and such. So how about for women in the same age group? So for women, major cause of death is cancer. So in the same age group, so it's in order of 25 to 30 deaths per 100,000 population, relatively constant in this age group over time. That's still true. And then accidents, you're less risky, I guess, is 15 versus, whatever, 50 to 60. But here's the red curve. So we come out of nothing and begin to get a read, goes up into the order of 5, and then decreases, as we saw with the males. So what do we attribute this change? I mean, certainly HIV remains a huge problem, but it's maxed in the, it's actually a slight decrease. Well, there are a couple of reasons, and I think you can put them into two broad categories. One is education, and the other is therapeutics. And how about education? So, in the mid-1980s, actually, I was at the University of Zurich in HIV, and my children went to a local Swiss school, which is kind of a traumatic experience for them when they were young. But all over in Zurich, you saw these signs. And this is a picture I took. You can see, this was like in the mid-80s, so 4, 5, 6 years after the discovery of the situation. Stop AIDS. And I think you can see it below stop. I can guarantee you didn't see these signs in the U.S. When I came back to UCSB, we're obviously chatting today, a couple of decades later. This is a picture I took on campus. I hope she doesn't have AIDS. I hope she doesn't have AIDS. Can't live on hope. It was later, you began to see this sort of awareness on campus. And I haven't seen one of these. Have any of you seen one of these? I haven't seen one of these for a while. So anyway, there's this education component that probably has contributed to the decrease. That's one aspect of it. And, what the public health folks suggest as part of the education in terms of limiting infections and spread is, of course, so public health, one, limit number of partners. Secondly, use appropriate condoms, as illustrated by the Swiss poster. And thirdly, as illustrated by behavior-wise bleeding, avoid intravenous drug use. Those are the 3 major recommendations of public health officials. All right, so that's the education side of it. How about the chemotherapy side, which is depicted on the slide on the right? So chemotherapy has been successful to a significant extent, and like education has contributed to the decrease. And so in chemotherapy, with the objective of chemotherapy, it sometimes is called antiviral therapy with antiviral agents, AVA, sometimes called. So the objective, and we touched on this a little bit, we're going to be visiting the subject in other instances between now and the end of the quarter, but the objective is to identify a viral gene product that's unique to the virus, like reverse transcriptase, or like the protease. And then you come up with an agent that selectively inhibits the activity of that protein without an adverse cytotoxic effect on some cellular function that you would need. Of course, if you destroy the host while destroying the virus, you really have gained much. So you need to strive to have an antiviral agent that's selective. Selective in the sense that we inhibit, since we're talking virology, the virus, and achieve that inhibition without being cytotoxic for the host. So that's been possible to an extent with HIV. And so the first inhibitors that were identified were inhibitors of reverse transcriptase. And the classic initial inhibitor that was identified, and in this case, we were inhibiting RT, reverse transcriptase, we're blocking the formation of the provirus. So if we can inhibit RT, we inhibit provirus synthesis. And the initial classic inhibitor was a compound called AZT. And what AZT is 3'-diazidyl 5'-deoxy thymidine. And the major drug brand of AZT, which is like a handout, is retinol here. So this is a pyrimidine. This is a pyrimidine analog. And so it requires activation. So you administer AZT, AZT gets activated in AZT triphosphate. And cellular enzymes catalyze the activation of the AZT triphosphate form. So cellular kinases do that. But once you've generated the triphosphate, then that targets the viral reverse transcriptase. And so what it does is compete binding of deoxy-TTB, the normal substrate, to reverse transcriptase. So the affinity for AZT triphosphate is about 100 times greater. So basically, you compete out the binding, you bind the inhibitor. So that's one mode of inhibition. The second is it's a chain terminator. So once the AZT is bound by the reverse transcriptase and it gets incorporated, it terminates the chain. So then you don't make the unit. All right, so that's AZT, and then there are a whole class of other compounds that were identified that essentially are chain terminators. They block provirus synthesis by terminating the DNA synthesis. That's one target. Then the second target that was really developed was the protease and inhibitors of the protease. And so, of course, if we inhibit the protease, the precursor GAG and the precursor GAG pol that get made can't be processed to make the viral gene products. So, if we inhibit the protease, we inhibit processing of our precursor polyprotein, whether they be GAG or GAG pol. And so there are the next, after reverse transcriptase, then the next wave of inhibitors were against the protease. There are many protease inhibitors now. But the major ones were indinavir and go to the idea. So this then led to something that's called heart therapy. And what heart therapy for HIV is, so it's H-A-A-R-P. Heart therapy, which means highly active antiretroviral therapy for the acronym. And basically it's a combination therapy. It's combination therapy that classically involves 3 drugs in the cocktail. And the drugs included 2 reverse transcriptase inhibitors, 2 different ones that work at different sites on the enzyme to inhibit it, and 1 protease inhibitor. And so the idea of combination therapy is that if you have multiple agents inhibiting, the likelihood that you'll get a mutation that in the target protein that then would make it insensitive to the inhibitor is much lower. The likelihood that you get 3 that would become insensitive to all 3 inhibitors. And this is an issue, on your handout I gave you, you don't have to memorize all these drugs, but there's lots of drugs, different ones that target reverse transcriptase or protease. And they're used in combination in heart therapy. Because what can happen is that mutations in the target can develop that then lead to resistance against the drug. And so in the therapy, what's really important is compliance in taking, maintaining the therapy, and then use of combinations of drugs so you wouldn't get resistance in all of the targets. In addition, though, to the protease and the reverse transcriptase, 1 and 2, in this combination therapy that's been kind of cornerstone therapy, there are new targets that have been developed. And the new targets target 2 additional viral enzymes, integrase. And so there are now FDA-approved integrase inhibitors, rolutegravir and elbicavir. These are 2 drugs that block the integrase. So if you escaped the RT inhibitors, you can target the integrase to block integration. If you block integration, you can have integrase. And then the most recent inhibitors are entry inhibitors. And so there's an inhibitor that targets the GP41 protein, the fusion activity. So there's a fusion inhibitor. And there's also an inhibitor that blocks the interaction of the chemokine receptor between GP120 and the receptor. The fusion inhibitor is approved, but I'm not sure that the binding inhibitor is yet approved. All right, so that's the direction that work is going, is to identify additional inhibitors that will no doubt lead to alterations in the combination therapy strategy for treatment. And of course, there's a tremendous effort to still get vaccines. But that effort is still ongoing, and we're not there yet. Okay. I'm going to change gears now, and we're going to move to our next virus panel. Any questions on HIV? Okay, so the next family of viruses, we're going back to viruses that have as a DNA genome double-stranded DNA genome, and these will be the herpes virus. Herpes viridae. Viridae virus family. Herpes comes from Greek to creep, and so it's creeping, spreading skin infection. That's where the name originally was derived from. So what are these viruses in terms of the human herpes viruses? So we're going to focus on human herpes viruses. But these are all envelope viruses. So here would be the schematic on the summary schematic that I gave you earlier. They're envelope viruses. They have mycosahedral nucleocapsid. Their size is in the order of 180 to 200 nanometers in size. They have, as I said, a DNA genome. It's double-stranded DNA. And so we're interested in human herpes viruses for our discussion. So on today's handout of figure 1, human herpes viruses have been classified into 8 groups of them, and the current nomenclature is HHV1 through 8. This simply means human herpes virus, whatever the number is. Okay, HHV1, which you have on your table, is going to be our prototype. This is herpes simplex virus, designated HSV1. And so this is the virus that's the major cause of fever blisters. There are oral, mouth, and ocular lesions. HHV2 is herpes simplex virus type 2, or HSV2. HSV2 is about 50% identical to HSV1 by nucleotide sequence. Classically, HSV2 is the herpes that's associated with genital and anal lesions. HHV3 illustrates a property of all herpes viruses. Many of us have been infected by HHV3 in the varicella form. What varicella is chickenpox. So highly infectious, typically childhood disease. And chickenpox is caused by a herpes virus, not a pox virus. The infection clears, and then, maybe my age group, we have reactivation, although it can happen earlier. And this disease is called zoster, and hence HHV3 in orange here is the VZV virus, varicella zoster virus. Zoster is varicella reactivated, and it's shingles. All right, we'll finish our characterization